All right, we ready? Okay. So, this one here is my answer. So I have the hydrogen on top. And I was walking around and check what you guys have. Some of you guys have the same setup as me, but some of you guys have a slightly different setup. Oh, not like that. So, this, for example, on this side is Xander's one. Now we know that Xander's definitely right when he makes the molecule because he's our canary. So, even though these are the same components, we have an amine, we have an alcohol, we have a chlorine, we have a hydrogen, there's no way I can rotate this molecule around to make the exact same molecule. So something's always wrong. So these two are matching, that's matching, but now these are the wrong way around. So let's go, okay, I'll fix that one. I do that, these two are matching, but now these two are the wrong way around. Yeah? If I do that, these two are matching, but now these two are the wrong way around. So when we are talking about optical isomers, we are talking about things that are mirror images of each other. So it's just a mirror image, that's it. So these are both the same answer, but different optical isomer versions of it. Okay? So when you guys get this on the assessment, I just want you guys to be mindful of how to draw it. There are different types of questions that they will ask. Some of the questions, they will just give you a nice, easy molecule to draw, and they'll have something that's like this, and they'll ask you to draw its two optical isomers. So for example, if I was to draw this in its three-dimensional shape, I would have carbon, I would have a hydrogen, I would have my chlorine, and remember we want to express 3D shapes, so we're talking about those tetrahedral shapes that we did last year. We have one coming out of the plane, which is my amine group, and then one going behind the plane, so I'm doing it as dashed, like that. Okay? If I want to mirror image it, so here's my mirror. I'm just doing the exact same thing flipped. So I do my carbon, I do my oxygen, OH like that. That guy's still coming out here, and H2. That guy's now over here, that guy's up here. So basically all I'm trying to do is I am flipping around these two. Keeping the same annotations for within plane, out of plane, or in the plane. So when I say this bold one, so if my piece of paper was right here, this is coming out of the plane. This and this are within the plane of the paper. So if I have the paper like that. And then this is in front, or behind, sorry. So we're thinking about three-dimensional shapes. So these two are mirror images. So there's nothing I can do rotation-wise to get these guys to be the same. That's the whole idea with the mirror image. So that makes them optical isomers. In order to have optical isomers, the main thing you need to have is something called a chiral carbon. So we need a carbon with 4 different things attached to it. So let me draw you guys an organic molecule, because they often will ask you, in this organic molecule, where's the chiral carbon? So that way you can draw the optical isomer. So let's say I had CH3, CH2, C, H, CH3. Let's say I have this molecule here. And we need to figure out, does it form optical isomers? What I'm looking for in this is I'm looking for a carbon with 4 different things attached to it. Four different things. That's the only way it forms. If I have 2 of the same, then I will be able to make the same molecule. So I'm just going to swap something out. Let's do the chlorines. And I'll go get hydrogen, just to show you guys what I mean. So in this case, I have taken my molecule, I have taken out the chlorine that was there, and I swapped it out for another hydrogen. So now my carbon has 3 different things attached to it, not 4. So when I have that, I am going to be able to rotate this around to make the same molecule. You see? So it needs to have 4 different things attached to it. If it doesn't have 4 different things attached to it, then I can move the molecule around so that way I have the exact same thing, which we know we couldn't do before when I had the chlorine. All right? So that's the first thing. So when you're going through this molecule, there's going to be spaces where it can't form, sorry, I should call that 4 different things attached to it. That is a chiral carbon. That's the vocabulary word you want. It's what we call that. So if I go through the molecule, I should be checking to see, does it have 4 different things attached to it? Because if it does, then it can form these optical isomers here. So this, for example, has 3 hydrogens and then this tail. So since it says 3 hydrogens, that's a repeat, so that one does not form chiral carbons. This one here has 2 hydrogens, a methyl, and then this branch attached to it. It has 2 hydrogens, so it doesn't have it's not a chiral carbon. This one here has a hydrogen, an OH, so an alcohol, an ethyl, and a methyl. That is 4 different things. So 1, 2, 3, 4. So this here is a chiral carbon. So that will be able to form optical isomers at that location. And then this here is out because it has the 3 hydrogens. So if I was to translate this into a drawing, I would do the exact same thing as above. Let me get another color. So I would put that carbon here in the center. It doesn't matter which one you put into what orientation. The only thing that matters is that you draw the mirror image of it. So you can put it any direction you want. So I'm going to put the OH up here maybe. I'll put the CH2, CH3 on this side. I'll put the OH on this side coming out. And I'll put the CH3 going that way. All right? So that's one version of that molecule. And then I would draw the mirror image of it. So we're just flipping. Oops, one of these should be dashed. I'm just flipping it around. So when I go to my chiral carbon, I'm just flipping these 2 around, but I'm keeping the same line drawing. So I'm going to draw my OH first. I'll draw, oopsie, did OH twice. It should be hydrogen. Put my hydrogen there. This is going to be the dashed line. Instead of that, it's going to be this. And then that goes here. That's my optical isomer. Questions, concerns, comments? No? All right, so some things to keep in mind is if you are able to draw one of the optical isomers, you get an achieved point. Congratulations. If you're able to draw both of them with a justification, that's usually a merit point. So the justification is that you need to have a chiral carbon, aka 4 different things attached to it, and then they often ask about its properties. So what properties does it have? So these guys will have exactly the same properties when it comes to melting point, boiling point, etc., etc. So these have basically the same properties. So for example, melting point and boiling point, solubility, all those things will stay the same. Reactions, that sort of stuff. The only thing that's different and the way we distinguish them is by polarized light. Distinguish with polarized light. So when we shine polarized light on these molecules, they're going to rotate in different ways. One's going to go clockwise, and one's going to go anti-clockwise. So one goes that direction, the other goes in that direction. And that's how we distinguish these 2 molecules. All right, they're more or less the same. However, they more or less are the same. They more or less do the same chemical reactions, so they have the same functional groups. But when we do talk about things that require enzymes, these guys can work very differently because we know that the enzymes are a very specific lock and key mechanism, that they need to have a certain shape. So if they're the wrong isomer, then it won't work in that lock and key. So if the reaction requires any sort of protein or enzyme, then the isomer does matter. But don't worry about writing that. The main thing you're just writing about is the general properties, and that the only thing you distinguish is with polarized light. Some fun facts for you guys is that there is, I heard this around the urban legend things. I think it's actually been disproven, but they said that the smell of lemon and the smell of orange are optical isomers of each other. I don't know how true that is. I've seen that pop on a lot, but I need to verify that one. I don't think it is. Would make sense, but I think it's just like a misfact or something. So you guys can look it up and check. I've seen it a lot on like images and stuff, and I thought that was a really interesting fact. But then I think someone else said no, it's not true. Did you guys find out? Yeah. I can talk though from a pharmacology perspective that these optical isomers are very important in drug development because sometimes with drug development, one optical isomer will be the drug and then the other optical isomer will not work or will be potentially harmful. So that is a very interesting fact about pharmacology is that these optical isomers can be very, very important in drugs because it may work, it may not work, it may cause problems. So they're very similar molecules, but they are mirror images, which can influence how they fit into various enzymes and things like that. Cool. That's all I have to do today for the notes. Any questions? Make sense? Hopefully. Should be hopefully an easy merit point for you guys. All right, let me walk you guys through what I recommend as your follow-up. So we've done our notes, we've made our molecules. You guys can take apart your molecules and return the pieces. Well, let me just explain your Mahi choice for today. So, options include you can work on the SciPy pages for the geometric isomers if you want. There's also some pages for the optical isomers. I just need to look up what page numbers they are for you guys because there's 2 pages in there. That's option number 1. Option number 2 is you can go to No Brain Too Small and do the collated questions on the isomers. Be mindful that if you're doing those questions, you will need to have the identification test worksheet that I gave you back in, I think, term 1, just to help distinguish what different functional groups are there, because it's not spec, but it basically acts the same as spec. The last thing I'll do is I'll pass out a CPR quiz if you guys want to work on that, which has the naming, the drawing, and the isomers. All right? Cool. So your choice, you don't have to work on it in that order. I want you guys to pick something that is at your level. So if you're struggling with the material right now, I suggest the SciPads. If you're feeling a bit more confident, then I say look at the CPR quiz or the collated questions on No Brain Too Small. Cool. All right, that's us.